Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment&Health. Eco-Environment&Health is a journal initiated by prestigious scientists with a strong editorial board, including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design, and broadcasting. The experts will accompany you like a harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision-making that deeply integrates informatics, ecology, and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access fee will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future.
So hello, Professor Sonne. Can you hear me?
Hello, Juan. So nice to meet you. Yeah, that's fully fine. Thanks a lot.
So what's the time there?
Oh, what's time here? It's just 3 o'clock in the afternoon, 3
In
p.m.
the afternoon. So you still have another half a day of working, right?
Yeah, something like that. And you will soon have to go sleeping, especially after my talk.
Oh, yes. But that will be too exciting for me to go to sleep immediately after your talk. So I see the polar bear as the background, right?
Yeah, right. It is exactly from Greenland. Yes. One of the species I will talk a little bit about.
Okay, that's great. So I'm very happy that you can join us in talking EEH. So that's a great help to us and to our journal.
Of course. And I must praise the introduction video. I liked it very much, and I can echo that the graphical department is really skilled.
So just follow us on Twitter, and we have a lot of interesting video and other interesting information about this journal and about stories.
That's great. I will go on Twitter afterwards then.
So I guess we can start. Okay. So first, I will give a short introduction to our journal and to you, of course. So hello everyone. I'm Huan Zhong from Nanjing University, China, and I'm the executive editor of Eco-Environment&Health. So Eco-Environment&Health, which is also called EEH, is an international and multidisciplinary peer-reviewed journal designed for the publication of the frontiers of ecology, environment, and health, as well as their related disciplines. EEH focuses on the concept of One Health to promote green and sustainable development, dealing with the interactions among ecology, environment, and health, and the underlying mechanisms and interventions. Our mission is to be one of the most important flagship journals in the field of environment health. And today, we are very happy to invite Professor Christian Sonne from Denmark to give us the interesting story. So Professor Sonne's background as a veterinarian, along with his wide and dedicated interest with the broad field of environment sciences, has enabled him to become one of the international leading scientists and authorities within Arctic wildlife toxicology. He is an expert in wildlife and human health, biology, and stress and ecotoxicology. He is an international renowned expert with more than 25 years experience in studying the environment in Europe, North America, and Arctic. So Professor Sonne has a unique ability of collaborating with scientists from many fields within environment sciences, and in that way, he has been able to produce novel and cutting-edge basic research within Arctic wildlife toxicology, research that also have very high applied and social value. His research has contributed significantly to identify the presence of toxic industrial chemicals in Arctic biota and to identify the harmful effects of these on Arctic wildlife, ranging from effects on the individual level to the population and ecosystem levels. Of course, this introduction is very short and impressive, but I don't think it's the full introduction to you, Sonne. So I think we need about 7 hours to tell your story, but I will give the time to you.
That was such a great introduction, Huan. Thanks a lot. That was, in fact, a really nice recap of a very broad field, which I have tried to put into a planetary perspective to give this talk about, the Arctic, about One Health, but also about planetary health, because it's one health, it's one globe, and it's one coherent ecosystem. I'll try to share the screen now. And please let me know, Huan, if you can see the screen I'm trying to sharing here.
Sure, I can see it clearly, and
Great.
I can hear you clearly.
Ah, that's great. I was not really sure about the 5G here, but so I'm very happy about that. So is it a full screen now, or is it a minor shot?
No, it's not full screen. Now it's okay.
That's what I was hoping for. So everyone, welcome to my talk, and thank you so much for being able to give this talk around this great EEH journal, which I appraise a lot, and I can only recommend to submit there. It's really efficient. It's really great review comments you get, and it's very smooth and very fast. So that's quite high quality. So this talk will be from molecular levels to planetary health, and I'll take, of course, the onset in the Arctic. It will start out with an Arctic perspective, because that's where I was born into science. And what is it with the Arctic? Why is it so important to understand ecological and ecosystem changes in the Arctic? That's because if you take a look at this fantastic paper from Nature, it's a great discussion piece about the climate tipping points of our globe. And if you take a look at this picture, this mapping at the left, you see that more than 50% of the global tipping points are either in the Arctic or in the Antarctica. And when you look at the Arctic and the Antarctica, there's no way that we can't see climate change. Climate change is now appearing 5 to 10 folds faster in the Arctic and the Antarctica. So you see that Arctic sea ice is melting, the Greenland ice sheet is melting, and the Antarctica is melting. And that will affect the biology and the climate and the ecosystem health of the entire globe. It's just like removing the rainforest in the Amazon. It will for sure be a tipping point, and maybe it will be a point of no return. So that's why we are watching so much about the Arctic and what's going on there in terms of climate change. But it's not just about climate change. It's also about biodiversity. It's about migration of species. It's about pollution. It's about new diseases, which is transported by invasive species into the Arctic, but not just around the Arctic. It's the whole globe. It's the whole ecosystem that's changing now, with new species coming in from new areas, transporting pollution and diseases. And these are some of these diseases are zoonosis. And I don't think I have to mention the COVID-19 pandemic. That's one of the zoonoses that recently appeared on our globe. And new zoonoses will come, and they may start in the Arctic. I'll get back to that in some later slides. So this redistribution of species and the loss of biodiversity has occurred over the last many thousand years, but it has for sure accelerated. If you take a look at the map or the graph at the right, it's from this great nature review. You should go and read it. Extinction has increased rapidly over the past, let's say, 5,000 years. And this is mostly human driven. It's driven by an ecosystem changes and ecosystem deterioration, so that species are simply going extinct or are redistributed. And that, of course, again, when we talk planetary health, that will be a redistribution of pollution and diseases as well. So it's not only about the species, it's also what they bring with them. And that will, of course, have a great impact at the molecular level and at the population level of different populations and of different species, and humans as well. And when we talk about the Arctic, the global goals apply all to the Arctic. It probably applies to most countries in the world, depending on what country you look at, but especially in the Arctic, there is so much ongoing these years in terms of climate change, in terms of pollution, and in terms of changes in society. So we have hunt, we have shipping, constructions, offshore constructions, you have tourism, you have all exploration and exploitation, contaminant transport, either locally or globally. I'll show that in the coming slides. There is a lot about fishery and blue food. The Arctic ecosystem wealth is in the ocean. It's not on the terrestrial environment. So whatever affects fishery and blue food is very important to the Arctic, but of course to the globe as a whole. Climate change is really affecting the Arctic far beyond, much more than we see on other places on the globe. The temperature is increasing in a magnitude of about 4-fold faster in the Arctic compared to anywhere on our globe. Then there is issues around food security in the Arctic, as everywhere else, but mainly because in the Arctic you eat raw tissues, you eat raw organs, you eat non-cooked food, even from polar bears and seals and toothed whales. That, of course, gives you the risk of getting diseases such as zoonosis, and also quite high contaminant exposure. So the global goals, it's about planetary health, also in the Arctic. So monitoring ecosystem health in the Arctic or anywhere else in a very remote ecosystem, it's not easy. It takes a wealth of a fortune. It's highly logistic challenging. It takes helicopters, it takes icebreakers. China have several, for instance. Canada also have several icebreakers also going to the Arctic and Antarctica, as have Sweden. and you have to go and take samples from either dead or live animals. That, of course, is a great challenge in itself. It's not just sufficient to take 1 or 2 or 3 samples. You might have to map the ecology of an entire ecosystem with maybe several hundred species from bottom to the very top, which is in the Arctic, animals like polar bears and of course, Inuits, humans. And you may have to analyze contaminants. You may have to analyze biogeochemicals like stable isotopes and stable amino acids. You may have to map diseases, etc. So it's a huge task. So when we talk about ecosystem health, One Health, and planetary health, it's not just something we do. It's a matter of collaboration between many different countries and many different scientific disciplines. And of course, we can only do it if we come together and try to shed more light on these planetary changes and how that affect the ecosystem and humans and animal health. And that is very urgent to do. And I think we are all aware of that. And that's also what this journal EEH is focusing on. In the Arctic, we're doing these One Health assessments over the past almost 40 years, focusing on pollution, focusing on biodiversity, focusing on human health, focusing on climate change, and also nuclear changes in the Arctic, and how that all this together might affect animal and human health, but in a very broad ecological perspective. So that's really why I like to apply what we've been doing in the Arctic for the last 40 years, that we should try and apply to ecosystems globally. And this is really the challenge of these decades. So just briefly, those of you that are not working in the Arctic, at the right, you have a figure showing how the Arctic is kind of a sink. The pollution in the northern hemisphere of our globe is literally raining into the Arctic, either through the atmosphere or through sea currents. So whatever is released in North America, in the EU, in China, Asia, wherever, is all transported into the Arctic. And I will give you some examples showing that East Greenland polar bear hunters and narwhal hunters in Greenland are the most exposed to mercury and to PFAS, fluorinated compounds, globally. And one of the substances transported is PCBs, and you're all aware that this is a thyroid hormone disrupting chemical, among other. It's brominated flame retardants, it's PFAS, and it's mercury. This is all transported into the Arctic. So you may think it's a very pristine ecosystem and that it is far away from emission sources, but in fact, it's among the most contaminant ecosystems in the world. And it's only contamination in areas like the Great Lakes and the Baltic Sea, which is lower. What is the problem? Yeah, here you have the example of a polar bear mother. She's trying to scratch the ice of a blowhole of a ring seal. She sits for about 2 hours, I think we were watching her, and afterwards she kills the ring seal. And is that problematic? It is problematic. It was not problematic 200 years ago. But mercury and also PCBs and all the other organic contaminants, it biomagnifies, as you know, into the top of the food webs where we have the toothed whales, the ring seals, and the polar bears. And all this is problematic, like for humans, because it's transported to the feces through lactational transfer. And when we talk contaminants, toxic thresholds are usually set via a lifelong exposure, but it does not take, for instance, lactational and in utero transfer into fetuses and neonates into account. And this is really where you have this transgenerational exposure and an extremely high risk of biological health effects, because it affects the development cell lines and the development of sensitive organs like brain, etc. So we have been using the polar bear as kind of a unique global thermometer for pollution, because pollution from the northern hemisphere is transported into the Arctic. So what we can find in terms of chemical analysis of polar bears is of great interest, not just to Arctic and to Arctic researchers, but it kind of mirrors what else is released into the ecosystems globally, or at least at the northern hemisphere. So they're at the top of the food chain, so they integrate health of the environment, and they, of course, contain a great wealth of bioinformation. That's bioinformation about pollution, climate change, diseases, including, of course, zoonosis. Zoonosis are these diseases that is transferred from humans to animals and back again. And then, of course, it's also about human exposure and translational medicine. At the end of the day, the humans could only survive in the Arctic because of polar bears, reindeer, and ring seals and toothed whales for food and for clothing, as you can see on this great picture from Northwest Greenland. And of course, polar bears attracts a great attention, not just because it's an iconic species, but namely because it contains a lot of information about the environment. So this is an example of a paper in Cell we had some years ago and recently in Science about how the polar bear can tell the story about the Arctic environment. This is again some data from polar bears. We have the longest time series of pollution in the Arctic in polar bears from Greenland. So this is for the past 40 years or something like that. And you can see that for PCBs, this is PCBs and different toxic thresholds. So you have a toxic threshold 1, 2, and 3. And despite the PCBs, they have been banned for many decades. We still see very high concentrations in polar bears, and they even see to increase because of a function of climate change and shifts in the food web. So the polar bears have access to new species that are much higher in contamination. Likewise for mercury in polar bear hair from West and East Greenland, it's red and blue here, is showing that you have a steep increase in mercury in polar bears. Despite that, we tried to, through the Minamata Convention, we tried to lower the emissions of mercury into the environment, but despite of that, mercury is released due to climate change, and there is ecosystem changes driven by climate change in the Arctic, so that we see a steady increase in mercury in polar bears, and thereby also humans, because humans eat the same as polar bears in the Arctic. Okay, this is an example about biomagnification of PFAS in the Arctic. This graph you really have to pay attention to. On the x-axis, you have the trophic level, and on the y-axis, you have PFAS in concentrations, but in a logarithmic scale. And you see when you go from the lowest, let's say lowest trophic levels like clams, all the way to polar bears, then you see a biomagnification of 30 millions in the Arctic ecosystem. There is nowhere in the globe where you have biomagnifications in these magnitudes. And on top of this, you have the Inuit hunters eating polar bears. And I will get back to that later. So this is about PFAS in polar bear brains. And it seems to affect, on the left, you see testosterone in a number of different compartments. 9 compartments we analyzed, and there seemed to be some positive relationships, so that testosterone in the brain of polar bears might increase as a factor of PFAS exposure, and that affects the behavior of polar bears, just like it has been shown it affects the brain of human kids in school, so they have more aggression and a more aggressive behavior. At the right, it's the dopamine system, which also seems to be upregulated, which has to do with motor functions and behavior and the vascular system of polar bears. So altogether, this reflects what's also known for humans, namely that PFAS in the brain of polar bears seem to affect hormones and receptors and different brain systems in a way that it might affect the ecology of polar bears. So more research is needed to look into this, because PFAS seems to be the new PCBs. Okay, this is unpublished. It's the first worldwide assessment about PFAS in human serum and the risk of immune deficiency or immune modulation. So overall, it seems that PFAS is affecting the ability to make or produce antibodies and thereby fight diseases. Could be COVID, could be any kind of disease. A recent paper suggested that there might be a relationship between high exposure to PFAS and the disability of fighting diseases like COVID-19, probably also through a lower response to vaccination. This clearly shows that Greenland, which is at the upper middle of the upper figure, is completely red, and showing that hunters, especially in East Greenland, but hunters throughout entire Greenland, is the most highly exposed on the globe, and then they are at the severe risk of immune deficiency or immune modulation, which is problematic because they have these high contaminant concentrations, but are also very much exposed to different infectious diseases because they eat all these raw tissues and raw animals. And that's why I suggest or propose or fear that despite it may not be so many diseases that are transplanted between humans and animals in terms of respiratory diseases, it might be other kind of viruses, but it could hint that just like for farmers where you have big concentrations of pigs or chickens, that this could be a hotspot for the risk of future upcoming or starting pandemics. So that's really something we should pay attention to. And why is that a problem? Reduced immunity? It's of course a problem because not just in the Arctic, but worldwide, we are exposed to a high number of different parasites. Some are listed here, and also some viruses listed here. And these are all found in both animals and humans, and can, of course, then be transferred forth and back. And I think around almost 70% of the diseases we have on our globe, they are zoonoses, meaning that humans and animals can affect each other. And then, of course, there is a number of bacteria, which is seen not just in the Arctic, but unfortunately also globally. And for example, in polar bears in Alaska, we have over the past 30 years seen a steep increase in some of these zoonotic diseases, and that can easily spill over into Inuit communities. And it might, if it's a highly transmittable, for instance, virus, it might kick off a kind of a pandemic, or at least some regional diseases that might affect societies in general. And of course, COVID is one example. I think it was probably starting in a bad, as I recall. So climate change is, of course, a problem. I don't know if you have seen this iconic picture from Svalbard, where a old polar bear male is either suffering from age and the disability of catching food and chewing it. They get a lot of tooth wear over age, just like humans. But of course, this was flagged up as being a result of climate change. And who knows, because we don't know the history of this specific male polar bear. But what we know, or what scientists have shown for Svalbard polar bears, is that their metabolome is quite different between spring and autumn. And that most really rely in their high-fat diet in spring, while they are mostly fasting in the autumn, where they really don't eat a lot. So one could fear that we might in the future see the autumn metabolome And that's where the polar bears are fasting. That's where their metabolism is changing. And that's something they can survive for some months, but it's not something that they can survive for throughout a whole year. So this, if this continues, it will for sure result in polar bears moving northward into the last Polyneas of the high Arctic, such as Canada and north of Greenland. And here you have a great Norwegian study about the metabolome and about the metabolism of polar bears, PPA and PPA-G, among others, and how different contaminants like PFAS, PCBs, and brominated contaminants, brominated flame retardants, and some chlorinated pesticides, how they might affect the metabolism of polar bears. So this is really a toxic cocktail. You have climate change, you have, which is of course affecting the metabolism of polar bears. Then on top of that, you have contaminants, long-range transported contaminants that likewise affect the metabolism of polar bears. So that's really a poisonous cocktail to these species. And we don't know how that will affect the population of polar bears in the future, but for sure it will. And as I said, the polar bears would probably have to move northward, and then at the end maybe just be in the polar basin and in the very high north of Canada and Greenland. We've also seen some genital deformities in female polar bears at Svalbard and in Greenland. We are not fully aware of what has caused this, if some is genetically inherited, or if some could be due to exposure to, for example, as seen on this figure, DDT and the metabolites of PCBs and maybe the total load of these different contaminants. But when we compare females without genital deformities with females which are having abnormal or genital deformities, we see that there is a higher concentrations of persistent organic pollutants in those females with genital deformities. So this has also been shown in laboratory mammals and also kind of in humans, that high exposure to these chemicals in utero or through lactational transfer may affect the development not just of the brain and the immune system, but also the reproductive system, which is of course a key issue when we talk population level effects. And what has caused this? Yeah, I have a good friend who has been looking a lot into the steroidogenesis, and that's what you see here. It's pretty complex. It starts out with cholesterol that you see here up in the white snow against the red building. That's then built into the production of prostatogens and androgens and estrogens. And upper right, of course, you have the corticosteroids, cortisol, among other, corticosterone in birds. And different species have different pathways. For example, the polar bear is a delta-4 pathway, meaning that they produce a quite high number of androgens during springtime, because that has to do with the induced ovulation in the females. But if you, for example, have a block of the CYP19, then you will have an up concentration of testosterone, because if you have a block of CYP19, the female will not produce estradiol. But then these testosterones in the females might affect their reproductive organs and likely also their reproductive ability. Then you have other pathways, such as the delta-4 pathway, which is, for example, sled dogs providing this delta-4 pathway or using it. And we often use sled dogs as kind of a surrogate species to study polar bears. So in terms of endocrinology of steroid hormones, maybe sled dogs are not the best candidate for studying as a surrogate species for studying polar bears. So that's also, of course, why you have to look deeper into this. But disruption of these different enzymatic pathways might affect the genital deformities in polar bears, which might again affect population development and a decline in reproductive capacity. Other species in these two different delta pathways are humans, alike to dogs, and then you have rabbit, mice, or rat, and hyena, similar to polar bears. And what's interesting about the hyena is that you see now and then similar, I would not say deformities, but similar macroscopic appearance of the genitals of the females, as we see in polar bears. So some of it might be evolutionary inherited, and some of it might be caused by exposure to these endocrine disrupting chemicals, for instance, like DDTs and PCBs, which you find in very high concentrations in polar bears. It also affects the thyroid hormone, so it seems that there is a negative relationship between PCBs and thyroid hormones in polar bears. That is also known from humans, unfortunately. And I guess we all know someone who's getting a supplement of thyroxine hormone simply because they are hyperthyroid and disease, which might be caused by a wealth of factors. It might be genetic inherited, it might be due to some chronic infections, and it might be due to age. It could also be due to your immune system, like you have diabetes, autoimmunity, and it could also be due to this exposure to these chemicals. And this is known to be a great problem in humans. And we also see that across a wealth of taxa, wildlife taxa, from birds to mammals. So something to really look into. Then, of course, we have the PCB and killer whale population collapse problem, where we have shown that PCB is extremely high in killer whales, much higher than in polar bears. And we have shown that about 50% of the killer whale populations will likely go extinct within the coming decades. And we see that especially at the Canary Islands and the Strait of Gibraltar, there's hardly any killer whales left. So that is another example of how PCBs might affect a wildlife mammal at the population level, just like we think it is for polar bears and PCBs, and the effects on the endocrinology, the thyroid hormones, and the ability to get pregnant. Another global occurring or reoccurring contaminant, that's mercury. I know it's not an enigma to any of you. There was recently a great paper out last year in Nature Communications, which was an assessment of the losses of IQ globally, and also the number of cardiovascular attacks or heart attacks and mortalities, and of course then the losses in economy in terms of IQ. And when you look at the right global mapping from this paper and look at the lower left map, which is showing where you have the highest mercury emissions, which occur in Europe and in North America and in Asia, then there's an overlap between the risk of these health effects and society effects from mercury at the right, which is closely linked to the emission of mercury in these different regions. So there's no doubt that the Minamata Convention is extremely important, just like the Stockholm Convention is for PFAS and PCBs and other organochlorine compounds. Then the Minamata is really important to lower the emissions of mercury and the risk to wildlife and human health. So this is really a matter of planetary health when we talk about mercury. And this is an example from Northwest Greenland. The example I showed you previously was from East Greenland, where people or hunters are eating, societies are eating polar bears. So there they are highly exposed to PFAS. This is about mercury, which is in West Greenland, where they eat narwhals. And in fact, I know this is a very, very busy figure, but on the x-axis, you have the year or the month, the weeks, and on the y-axis, you have the mercury concentration in hair, like head hair from Inuits, or was it beard? I think it was beard. And it's showing for 1, 2, 3, 4, 5, 6 persons that they have quite high concentrations of mercury, especially in the autumn where they eat a lot of narwhals. That's around week 37. You see that the concentration of mercury is really increasing before and after that period. And some of the hunters are more than 20 times above the EPA guideline, which is just around 2 ppm. So this is showing that the highest exposed humans globally, that's in the environment, that's hunters in Northwest Greenland. The green person you can see just close to zero at the y-axis at the very bottom. That's one of my colleagues from Denmark, and he ate some narwhal in week 23, 33, and 35 and 37, and you can clearly see that he is having a steep increase in mercury in his hair. So eating narwhal is not something you should do on a daily basis. You probably need some supplement of antioxidants and selenium in order not to die from this if you eat it on a daily basis. So this again is about planetary health, because all this mercury is long range transported into the Arctic. There's of course some bedrock mercury in the marine ecosystem, but the majority is transported or re-emerging from melting glacial ice. And we have shown that mercury in West Greenland narwhals is increasing due to industrial releases and climate change, and due to changes in the food web dynamics. So it's transported more and more into the high Arctic, and then there is a food shift for the narwhals, which increases the mercury concentrations in these iconic whale species being eaten a lot in the Arctic. And this was based on annual growth layers in the tooth of the whales, which you see at the lower left in figure B. So another example that mercury is increasing in the Arctic. And mercury is also known to be both an immune and neurotoxic substance. At the left here, you again see the concentrations in a polar bear brain. You have the mercury concentrations on the x-axis. Then you have another NMDA receptor in the brain of the glutamate system in polar bears, and you can see that there might be a negative relationship, so that the high mercury concentrations may lower the activity in subparts of the brain, and thereby affect the behavior of the polar bears. On the upper right, you see how mercury is decreasing the ability of the lymphocytes to proliferate or to divide. And since the lymphocytes recognize antigens and produce antibodies, high concentrations of mercury is known to affect the immune system across a high number of species, including seals and whales and polar bears, but also humans and also laboratory animals. So mercury and PFAS and PCB in the environment is for sure a great problem globally, not least in the Arctic. So summing up, if we should sum up what's so special about the ecosystem in the Arctic, is that there is also some resilience in the Arctic. And that has to do, of course, with we know that being outside in the environment affect the human physiology and mental health to an extreme value to be healthy. So, and Inuits are outside in the environment day long, but it's also a matter of marine food is containing a lot of good stuff, so to speak. There's a lot of antioxidants, there is a lot of selenium, and there is a lot of polyunsaturated fatty acids, and all this is to great benefit to the health in terms of cardiovascular diseases And also in terms of your immune system, it's stimulating your immune system, among other. So that's the resilient part. Of course, then in the fragility part, you have pollution, you have exposure to infectious diseases, and you also have new tipping points such as shipping activities, and of course, not new, but some decades about climate change, which is really affecting the health of the Arctic ecosystem. And again, just to recap my two first slides, if and when the Arctic is affected this much by climate change and pollution and diseases, it will affect the whole globe. It will affect the climate of the whole globe, and it will also affect the disease patterns of the whole globe, because melting of Arctic sea ice and redistribution of species into the Arctic and out of the Arctic is going to change our world, and it's going to change the composition of our ecosystems. But it's also going to change the transportation possibilities through the Arctic, and then will of course affect the Arctic biodiversity to a high degree. So the resilience, of course, you have marine protected areas, and you have different international organs like the IUCN providing with specialist groups and also the global goals. But much more in terms of collaboration and research is needed to really understand how planetary health and how different global ecosystems are affected by these anthropogenic stressors, and of course by stressors and humans' influence in general. So I think that was it. I think I should talk in 40 minutes, and I think I was talking in 42 minutes. So now it's time for questions. I think thanks a lot for listening.
So thanks, Professor Sonne. So that's a very interesting talk. And I just asked our staff, and they give me the number. So now we have about 1,500 people listening to your talk.
But that's amazing, Huang.
Yeah, because we have several platforms.
That's amazing. That's a great thing.
Yeah. So and we got another questions for you, and I pick a few from them because I don't think we can have the time to answer all the questions. The first question is that we know that polar bear is at the top of the Arctic food chain, and theoretically, polar bears would accumulate high levels of methylmercury. Is there any studies reporting the level of methylmercury in polar bears and the associated neurotoxicity in response to climate change?
That's a very good question, and I think when it comes to neurotoxicity and climate change and polar bears, I think the studies I have seen have either dealt with mercury exposure and climate change in polar bears, so food web changes in food web dynamics. But I don't think I have seen any studies that has combined changes in food web dynamics driven by climate change and mercury exposure and effects on the brain in polar bears. No, I don't think so. That should then be a kind of a geographical study where you compare different polar bear subpopulations. Some are high and some are low exposed, and then brain biomarkers. But I think that has only been done in Greenland and Canada. So no, not so far to my knowledge. Good question, though.
Okay, thank you. And the second question is about PFOS. So the question is, why the bioaccumulation factor BAF of PFOS be much higher in Arctic compared to other areas?
That's also a very good question. When I talk to the biologists who are experts in this and who has been working in the Arctic for decades, Rune Dietz among other, it has to do with that the species are slow growing because it's cold. And when you're slow growing, you're maybe not that big, but in fact, you're very old. And that seemed to increase the biomagnification in the Arctic ecosystem. Then on top of that, you have the polar bear, and on top of that, you have the Inuit, which is adding even more to the biomagnification of the ecosystem. So if you can compare with the terrestrial ecosystem, then you may have the biomagnification factor, which is maybe 10 or 100 when you go from soil to plant to cow or pig or what you go to humans. When you compare with other ecosystems, often you have maybe toothed whales and seals, but then it stops, and polar bears and humans really add further to the biomagnification in the Arctic. But it has to do with slow growing organisms.
Okay, thank you. And the next question is also related to this. The question is the food chain longer or shorter in the Arctic? And how the climate change will affect that?
That's a very good question as well. And maybe I should also have re-emphasized that when we have these extra trophic levels, it's of course also a longer food chain, which is also adding to the explanation. If climate change will affect either increase or decrease the PFAS accumulation in the Arctic, we don't know. But if we look at PCBs, for example, we can see that when it comes to polar bears, we can see that polar bears now have access to seal species that are several trophic layers above the ring seal, which they are usually eating. And that is probably driving and explaining the increasing concentrations of PCBs in polar bears, despite that the global levels should decrease. And that has been investigated using fatty acid signatures. Did I answer that question? I'm not sure.
I think so. Yeah, thank you. So the next one is a very open question, and it should be a very interesting one. So the question is, will the melting of glaciers be a potential source of contaminants and pathogens? And is there any confirmative evidence? Is it an important source compared to the anthropogenic source?
I love that question. That's something we discuss a lot. So let's start with mercury. There seems to be a re-release of mercury from melting glaciers in Greenland, for example. The same has been seen by lead, which you have leaded petrol globally, which is now history. Then you can see in ice cores that lead has increased in the Greenland ice cores, and then it has decreased again. When it comes to mercury, yes, it can be released from melting glaciers. We have shown that in East and West Greenland. But what is maybe even more interesting and important is that it's also released through release of terrestrial dissolved organic matter, DOM. So if you have we don't have that in Greenland, but if you look at Svalbard and other areas where you have large tundras, then the release of DOM will release very high concentrations of mercury, and that has been shown in a great paper in Science some years ago. So when it comes to mercury, definitely we will see a very large release. When it comes to the organic, persistent organic pollutants, I'm not sure it's the same. That has maybe rather to do with invasion of new species and changes in food web dynamics, so that you will, as a polar bear, get either lower or higher exposed.
Okay, that's a good answer. So the next question is also about the polar bears. So the increase of mixed mercury levels in the hair of polar bears, as you show in the presentation. So what's the reason for that besides the increased release of contaminants of mercury into the environment? So what's the other reasons driving the increase of mixed mercury levels in polar bears?
So that has to do that despite we have the Minamata Convention, there is still a lot of fossil burning, and there's still a lot of concrete production and small scale mining in Africa and South America, for example, and that releases a lot, and you won't believe it, but that releases and I didn't show it. I have a graph if someone want to see it. That releases a lot of mercury into the atmosphere and into the environment. Aside from that, climate change is re-releasing this, and also El Niño and other volcanic eruptions is also adding to the increase in mercury, in probably also in polar bears. We are we have a new project and a new ICP-MS which can analyze for mercury isotopes. So this is the question we will look into. Where does these isotopes come from, if it's possible? And then hopefully we can then pinpoint why the concentrations has increased in polar bear hair. That was both West and East Greenland. If you look at some of the Canadian polar bear subpopulations, the increase is even steeper.
Okay, so you already answered the half of the next question, because the next question is there any studies using the stable isotopes of mercury to explore the changes in the sources of Miss Mercury to polar bears?
That's a fantastic question. I'm happy. These are brilliant questions. People out there know a lot about the Arctic, I can hear. And it has been, when it comes to mercury isotopes or stable isotopes, they are not easy to analyze. You need a quite high concentrations and sample size, and you also need to understand the patterns that you end up with. So it's kind of mapping the ecosystem. We try to map the ecosystem at different trophic levels in Greenland at the moment, and through that we try to understand the mercury stable isotopes and how that might explain the different sources, just like we do with fatty acids.
Okay. So although we also have some other questions regarding to your presentation, but I don't want to use all this time for answering this question, because we have something more important to do. So many people ask you and want to take this opportunity to ask you, as we know that you are the chief editor of many journals, and also associate editors and the board members of many known journals, and you are a great writer for papers publishing in like Nature and Science and other top journals. So can you give us especially some suggestion to the young scientists or early career scientists in China and in other parts of the world? So what's the secret of writing a good paper? I know it takes you about hours or days to tell all the tips and skills in writing, but if we only have several minutes, can you give some suggestions? And this is also the bonus to the people who listen to your presentation today. Thank you.
Of course, that's also a great input. And I must admit that it took me 20 or 25 years to try and understand how you make up a great paper. And people are different. Some people like very long, detailed papers, and others like some papers with great highlights and then a great supplementary. So it's a matter of, if you ask me, it's a matter of starting low and then build up your network, and then try to read the papers or the journals you would like to get into. Then try to adapt that writing style. Read many, many, many papers. Maybe you won't recognize that you then understand how they are built up, but it will for sure be saved, filed in your brain, so that when you start writing, you will try to adapt to it without really knowing it. But never start with Nature and Science. If you ask me, I would start at some relevant journals. Read a lot of papers. If you're in doubt and you want to submit the paper, write to one of the editors and then directly ask, is there some pitfalls I should know about before I submit? Some pitfalls that is maybe not easy to read on the journal's homepage. It could be for environmental pollution, only have one corresponding author. But read a lot of papers, but don't start out high. Read papers from a journal you really like and read a lot. Try to adapt the writing style. Try to understand how you compose the paper. And then I have learned from my many, many mentors, never be afraid to ask others to help you to write the papers, because you learn so much. Don't sit with it day and night for half a year and show it to someone, and then you get depressed because it's painted all red and yellow and with tons of comments. So get help from the very start. Read a lot of papers from a few journals so you understand what they would like to see and exactly how to compose the paper, what should be where. And then don't write too much. Try to be storytelling. It's not a matter of being nerdy and very detailed. Sometimes a paper is better if it's explained in elegant, easy understanding sentences. Then put all the details in the tables, in the figures, and in the supplementary, because it's like reading a thesis. If it's too long and too complicated, you get very tired as a reviewer. Maybe that was kind of. Never hesitate to write me an email, and I can help them. When I always do that. And it's not to be a co-author. Ask me if you want to publish in environmental pollution. Write me an email, and I'll just give you a few guidelines about what to do and what not to do. Was that an answer?
That's a very short but very informative answer, I think, because we all learn a lot from your writing, and that's really fascinating to read your papers, especially those short ones you publish in Nature and Science. And I think the last question about writing is how to keep the balance between the high novelty, high importance sentence and the scientific details you have to tell your audience. Because sometimes, even for myself, I think it's a very hard thing to do.
And when, to be honest, it's exactly the same for me. If I write something, I have co-authors, it's all painted red and blue with tons of comments. I receive exactly the same responses as a young student would, but maybe in a different way or at another level. But I can also be depressed by co-author comments or by reviewer comments. So sorry, Juan, what was your question? It was about.
So how to keep the balance between the details and the novelty and importance, the short sentence.
Of course. So the short sentences, one, it depends on the journal. Science of the Total Environment, you can give all the details you want, as long as you have a great story and a great writing style and some nice figures and tables. When it comes to high impact journals, don't give too many p-values or don't give too many statistical values in brackets. Then you can give maybe highlight a p-value or highlight an interesting modeling result data somewhere, but don't do it all the time. Don't give a p-value in each sentence in the results section, but do it only for specific highlights.
So this goes back to your first suggestion to read as many papers as you can for the journal, right? So, okay, so we already have 1 hour. So I know we all know that you're very busy, and thanks so much for sharing us with this in this 1 hour, and we get a lot of information from you. And we are also very glad to hear your interesting story about polar bear. So that's a fascinating night for us in China here. So again, thank you for joining us in Talk and EEH, and thanks for submitting your papers to EEH and publish with us. And we hope that you could visit us in the near future in China and in Nanjing. So
I would
good afternoon.
Thank you very much. Thanks for inviting.
Bye-bye. Thank you.
Bye-bye.